Our next speaker is Beth Mormino of Stanford University. The title of her talk is Cognition and Amyloid in Healthy Adults, Who is Resilient and Who Declines?
Okay, so we've already talked about this a lot, but I'm going to quickly go over the concept of reserve and Alzheimer's disease. I'm then going to discuss, spend most of the time talking about amyloid-positive, clinically normal, older adults, which are both an opportunity to investigate early Alzheimer's disease processes, but also, just as important, the factors that influence reserve. I'll present data looking at longitudinal decline in amyloid-positive normals and end with some specific examples of amyloid-positive normals that seem to be resilient to cognitive decline despite this pathology. So as discussed earlier, the concept of reserve in Alzheimer's disease, one definition is really a disconnect between the pathological underlying burden and clinical function. So this is depicted here. So for a given level of pathology, a low reserve individual will have more severe expression of the disease clinically than a high reserve individual. If we superimpose that on the biomarker cascade of Alzheimer's disease, as shown in this plot, we can think of this. So in this model, we have amyloid accumulation happening very early, followed by a number of downstream changes, such as accumulation of tau, and also changes to neuronal integrity. So if you think of this in the reserve framework, we have amyloid here, and reserve may just shift the time delay between when amyloid accumulation begins and when we actually have a clinical expression of the disease. However, it's also possible that these factors that contribute to reserve have a direct effect on pathology. So we haven't really talked about that today at all. So we know that synaptic activity actually has an impact on the release of amyloid from neurons. Animal mouse models of AD have been shown to have less amyloid plaques when in an enriched environment. And even in human studies, higher reports of lifetime cognitive activity have been associated with lower levels of amyloid detected with PET. To tie these together, we have argued in the past that an enriched environment, which is likely a contributor of reserve throughout the lifespan, may actually influence neuronal efficiency, which ultimately may reduce amyloid late in life. So it's important it's very complicated, but these different mechanisms that underlie reserve may both interact with the pathology itself, but also how you decline when faced with pathology. So given that reserve is probably relevant throughout many different points in the Alzheimer's cascade, neuroimaging really provides a unique opportunity to directly investigate these specific associations. So with Alzheimer's disease specifically, as I mentioned earlier, we think amyloid is this early event. Tau is also this early event that begins in the medial temporal lobe, and one idea is that the spread of this pathology into neocortex is accelerated by amyloid. Downstream to that, we have changes in neuronal integrity and eventually cognitive impairment, and it's possible that these mechanisms may influence any point of this cascade. And so we can measure this using neuroimaging, so we can measure amyloid with amyloid PET, more recently tau, both confined to the medial temporal lobe, and also the spread into neocortex. Multiple imaging modalities are available, which we've heard about some earlier today, to assess neuronal integrity, such as structural MRI and also functional MRI. So my work has really focused on amyloid-positive, clinically normal individuals. So here's a meta-analysis looking at both postmortem and amyloid imaging studies, documenting the prevalence of high amyloid despite being clinically normal. The prevalence is on the y-axis and age is on the x-axis. Multiple cohorts are represented. Each bubble is scaled by sample size. And what we can see, there's little evidence of high levels of amyloid before age 60, but after age 60, there's this linear relationship such that about 30% of people in their 70s will have high levels of amyloid. So investigating these amyloid-positive normals, we definitely see these very subtle AD-like differences in the brain, both in terms of structure and function. So here's work by Alex Becker and Keith Johnson at Mass General showing reduced gray matter thickness in the posterior cingulate. This is work I did with Bill Jagus during graduate school, showing reduced connectivity in the default mode network, a network that's known to be vulnerable in Alzheimer's disease. Many other people have published similar findings, showing at the group level there are certainly subtle differences in amyloid-positive, clinically normal older individuals. This is recent data from the Harvard Aging Brain Study. The Harvard Aging Brain Study is a study of clinically normal individuals. We have 277 subjects in this study, all have been scanned with PIB-PET to give a measure of amyloid at baseline, and they've been followed for about 3.5 years longitudinally. So we also find evidence that the amyloid positives do in fact show longitudinal decline over time. This is most pronounced with episodic memory. The amyloid positives are in the red line, but we even see group differences in other domains, such as semantic memory and also executive function, just to a lesser degree. This pattern that amyloid-positive individuals show cognitive decline over time has been published over multiple cohorts now. It's very consistent finding in the literature. Also, another consistent finding is that it tends to be across multiple domains. It's not necessarily specific to episodic memory to decline. This has been very consistent. Less consistent is the cross-sectional associations with amyloid and cross-sectional performance. So even though at the group level we certainly see that these amyloid-positive individuals do show decline, there's a tremendous amount of variability in this group. So this is the Harvard Aging Brain Study plotted again. Sorry, the plot doesn't look that great. Here are all the amyloid negatives, here are the amyloid positives, and you can see there's a tremendous amount of variability in the slopes. There are some people that are declining, other people that remain normal or even improve over time. So one thing that I'm really interested in is what factors explain this variability, and this is very relevant to the concept of reserve. So one obvious thing is tau. So recently, since we have been able to measure tau in vivo, we can ask this question. Here are some example cases. Here's an amyloid negative normal control. Their PIB scan is right here. It's very negative. They also have a pretty negative AV1451 scan, although we do see some signal in the medial temporal lobe. Here's an example of an amyloid positive scan with distributed PIB signals, positive case. We also see this initial spreading from medial temporal lobe into inferior temporal cortex that's being picked up with this tau scan. This is in contrast to an Alzheimer's disease patient subject who has very distributed levels of amyloid, which would be hard to differentiate between the normal and the Alzheimer's case, and also more widespread tau involvement throughout cortex. We've scanned about 130 people with tau PET within the Harvard Aging Brain Study, so not the full sample, but about half. And what we do see is that, so this is memory slope on the y-axis and tau from the inferior temporal on the x-axis. The amyloid positive folks are shown in red, and we definitely see that the people with the most decline in our group are those that are both amyloid positive and also have high levels of tau. So this is certainly one contributor to the heterogeneity in the amyloid positive group, which is what we hypothesized. So another thing that seems to explain variability, which it's interesting, it's been brought up a few times so far in this conference. So we actually see more decline in the females than the males when we stratify by amyloid. So here's Here's the plot I just showed you a few slides ago, showing just the amyloid effect on memory, semantic processing, and executive function. The amyloid positives are declining across all those domains. When we break the group into females and males, we actually find that this amyloid-related decline is only present in the females. We actually don't see this decline in the males. So in our hands, it does seem that the females, when faced with amyloid, do seem to be declining at a faster rate than the males. Another factor that seems to influence this decline are genetic risk factors, and this is just a few examples. I think this is really just the tip of the iceberg when it comes to genetic predictors of decline within the amyloid-positive group. We published a few years ago that ApoE4 has an additional effect above and beyond amyloid on decline. So here are amyloid positive and negative, here are the amyloid negative groups. Here's the amyloid positive group, but E4 negative. Here's the amyloid positive group that's ApoE4 positive, and we see that the most decline is present in this group. So although we know that ApoE4 is a risk factor for having high amyloid, it also seems to act synergistically to influence decline. This is work by Yen Lim and Paul Murup from the ABLE study, where they additionally separated people based both on APOE and also BDNF, and found that people with the 2 risk alleles were actually the ones driving the greatest decline. So this is another example of genetic risk factors influencing decline in the face of amyloid. So something we've recently begun to look at is individuals that have been followed for a long time. So I showed this plot earlier. This is in HABs, showing the tremendous amount of variability. This is the amyloid negative folks and the amyloid positive folks. They're only followed for 3.5 years. There's this interesting group of subjects that entered into the ADNI study during ADNI 1 that have been followed for a very long follow-up. So this is over 7 years of follow-up. It's only 50 subjects that had CSF amyloid at the beginning. But we see a similar pattern. So there's definitely this group level effect, such that the amyloid positives at the group level are showing decline. But there's a lot of variability, such that some people are declining and some people, even with a very long follow-up, are remaining remarkably stable. So a nice thing with ADNI is that they do repeat CSF measurements. So one obvious explanation is, is it just a misclassification? So there's something wrong with the test at baseline. So when we look at amyloid over time, that actually only affected 1 case. Everybody else remained amyloid positive through 5 measurements or so over time. There's this one amyloid positive case that seems to do really, really well. They really jump up. So that was actually a misclassification. So we could toss them out. But even within the remaining cases, there are definitely cases that seem to be very resilient to their amyloid, even after being followed for 7 years. So I'm going to focus in on two of these cases. So it's 2 of the 12 cases that were amyloid positive at baseline that were followed for 7 years. This is case number 1. So this person, has dramatic practice effects. Actually, these are related to version issues, but they're definitely doing better over time. This is the blue dot here. So this is showing CSF tau by A beta. So this would be the quadrant that's 80-like, so low amyloid and also high tau. So this person falls smack in the middle of this very 80-like pattern in terms of the biomarkers. The red line has a very slight decline, but is doing very well and staying normal. This person is amyloid positive, but does not have evidence for elevated tau. So this individual, the blue individual, this is a male, 72-year-old, has 19 years of education, so it kind of fits the classic high education example, is also a 3-3 in terms of APOE. He had visits for 10 years between 2006 and 2016, actually became MCI in 2014. So if you look at this again, you can actually see this decline at the very end, but it's just a decline to the person's baseline. So this is kind of a classic example of the high reserve masking. This person's still doing very well in these tests. And it doesn't, from just looking at the test, it's not obvious that this person is progressing along the trajectory. This case I find just extremely fascinating. So this is somebody who was 79 years when they entered the study. It's a female, only 6 years of education, and has an E4 allele as well. And this person remained normal at every visit. So this person, they have multiple risk factors: low education, an E4 allele, amyloid, consistent amyloid. They don't seem to have high levels of tau. So it brings up the question, is there these additional protective features in this case that are preventing them from progressing along the cascade? I think these 2 case examples are really interesting because first, they weren't that hard to find. It was more than 10% of the sample that was available, and it also implies that the mechanisms underlying their resilience are completely different as well. So to conclude, reserve likely affects the Alzheimer's cascade at multiple points along this trajectory, and I think neuroimaging is a very powerful tool to try to tease that apart. Alzheimer's disease pathology is common in normal aging, and really gives us this opportunity to understand these mechanisms that protect against Alzheimer's dementia. And I find it very difficult to disentangle this impact of this very early pathology from normal aging. It seems like we need to really consider these things together. And although amyloid positives at the group level definitely show decline compared to their amyloid negative counterparts, there's tremendous heterogeneity that exists at the group level, and there's very basic demographic factors that might explain this, but a tremendous amount of unknown factors that are still left to be discovered. And I do find, this theme has come up a few times already, this idea of an escapee, these outlier cases. And I think we're going to be able to do this more and more with these studies that have biomarkers at baseline with really long follow-up, and isolate these people who have remained cognitively intact despite underlying burden, and interrogate these subjects to identify protective factors. So hopefully that's something that I think we could do as a field moving forward. So I want to acknowledge the investigators at the Harvard Aging Brain Study, in particular, Risa Sperling, Keith Johnson, and Doreen Rentz for their help on this work. Thank you.
Thank you. Questions?
That was a great talk, thank you. So pathology studies have shown that tau mediates the relationship of amyloid with cognitive decline. Are you seeing that in the in vivo data, or? Yeah,
so I showed the one plot I showed with tau. It definitely suggests that it's really the amyloid positive people that also have tau that are driving this. And oftentimes when we put both, when we do the classic mediation models, if we put both amyloid and tau into the model, right, it's tau that's related and not amyloid. So
amyloid becomes non-significant after accounting for tau.
Yeah, typically that's what we see. I think so I think that's consistent, with what you guys have published. Right. I think the unique opportunity here is to understand the factors that promote tau in the face of amyloid. So right, there's, most of the amyloid positives don't have extensive tau, within this clinically normal range, and it's to be determined whether or not all these amyloid positive normals will develop tau, or if there's these protective factors that might block that process.
Yeah, great, thank you.
There's been a recent study where they developed a risk score for Alzheimer's disease, looking at multiple genetic markers. Do you have that kind of data on these people, and have you thought about looking at that to see?
Yeah. So Raul Desikan just published, these risk scores. So we're starting to look at that. So we actually published a paper over the summer doing this in ADNI, so looking at polygenic risk of cognitive decline, and we weren't able to look at with respect to amyloid status. But I think that's definitely something that we're interested in. Whether or not we have the power to do this. I don't know with just halves, but I certainly think that that's going to be a powerful genetic risk factor potentially.
Thank you. Okay, so 1, 2, 3, 4. Yakov, you're 4.
Okay, so very nice talk, thanks. So you show very nicely, in the A beta positive, tau positive, you see decline, and also in the present E4, you also see similar phenomena. The question is, do you know any interrelationship between this tau positive and the E4? Is the E4, independent effect, or E4 goes through the tau, or any relationship there?
Yeah, so the Rush group has published some of these path analyses showing actually some direct paths between ApoE4 and tau, both in the medial temporal lobe and also in cortex. So we're beginning to look at that. It's a very small sample with just, we have 140 normals that have been scanned with PIB, with tau, only 30% of them are amyloid positive, and then you break them up by ApoE4 status. So it's complicated, I think, to get at this, these 3 variables working together in this sort of data set. But that is certainly something that we're going to look at, and I think there's already evidence of this, that there is this, it's a smaller path, the ApoE to tau path, than ApoE to amyloid, but I would suspect that some of this ApoE effect is redundant with tau.
Thank you.
So this is a little out of the box, but have you ever considered looking at chronic traumatic encephalopathy? Because 50% of the players are A beta positive, and almost all of them have tau. And there's probably a cohort who are cognitively normal, but yet have a high burden of this physiologic disorder. And so that might give you insights in a younger population about, cognitive reserve or what it is that maintains their cognitive function in the setting of such severe pathology.
Yeah, so I know people are doing these sorts of imaging studies in CTE. I think it is to me it seems like a slightly different question though, because you're asking about pre-existing a pre-existing disease and what happens when you put amyloid on top of this. So very similar to, we know that if you have amyloid and also stroke, you're going to do less well than if you just have amyloid. So I think it would, I guess, one potential question is the tau and CT, is it, in particular, does it have a response to amyloid? So I think it's definitely an interesting direction. I know there are studies that are being done in this population.
Okay. Can we have short questions and short answers? I think you're next.
Well, I was just going to ask,
Go ahead.
I know there's been some fMRI done in this population as well, and that it's greater, hippocampal fMRI is greater in amyloid positive, correct?
Well, so that's been a little inconsistent. So Bill Jagus presented some fMRI work, earlier today.
Yeah.
In HABS and also, in the Berkeley Aging cohort, we have looked at fMRI. There is some fMRI work from the Dallas Lifespan Study of Aging as well, where I think, if you actually do look across all these different fMRI studies, there are some slight differences between them, but also different tasks.
Yeah, well, what I was going to ask you is, do you have any tau, there's the experimental animal studies indicate that that activity drives the tau.
Yeah, so we are starting to look at this, the connection between.
I mean, I think that's more interesting than, yeah.
Yeah, so that's definitely being collected right now. It's definitely in HABS, I'm sure, also in these other groups as well.
Okay, thank you. Thank you very much, Beth.